Hi everyone, happy Monday. Uh, Today it's an exciting day because we are going to talk about microbiome, and the plan is that we understand how are they important to us, what are their functions in our human body. Then we talk about what happens if there is an imbalance between some of the good bacteria and the bad bacteria, how they interact with the gut epithelial layer, and how that aspect is now associated with various chronic conditions as well as some autoimmune conditions. Then during the latter half, we'll try to kind of revise about fiber. I'm hoping you would recollect from your previous subject about the functions of fiber, types of fiber. But today, our main emphasis would be to understand which are those prebiotics and probiotics that support the microbiome in our gut and thereby help in modulating or preventing and even correcting certain chronic diseases. My aim is that kind of we can finish around 15 minutes earlier, and then, uh, I can break you into breakout rooms so that you can discuss few things among yourselves. All good? Sounds like a plan.
All good.
Thank you. Okay, so human beings have always been curious. So we want to know about ourselves, we want to know about the universe, Mars, black hole. Uh, and this curiosity led to this revolutionary expansion of knowledge that there are um, some bacterias living in our gut. This term microbiota uh, actually replaces, remember when we used to study biology in our school, we used to talk about fauna and flora. So in a way, this term is now preferred, microbiota over flora. And it actually includes non-bacterial elements as well, such as viruses and fungi. But since the discovery of bacteria in our gut, we have been able to gather evidence only in regard to bacteria. So that's why today's lecture and even current microbial fecal testing that help us devise nutritional intervention for people suffering from dysbiosis, it all relies on research on bacteria. So in the human gut, as you can see that there is some extra info there. The current understanding is that we have more DNA of bacteria than our own. Okay. And usually when we are saying microbiome, we are referring to that genomic content of the microbiota. So they are not only present in the gut, they are present on skin, respiratory tract, vagina. There is a whole new research area that is looking at vaginal microbiome and how it can impact a mother's health and even baby's health. Now, earlier, we used to believe everything is sterile, like placenta is sterile. So the baby, when comes into being, when a baby is born, everything is sterile. But the development of the gut microbiome actually begins in utero. So a group with Agard, it's the name of the scientist, and his colleagues, they all actually looked at the unique placental microbiome niche. Has anybody seen placenta? Sorry, I'm digressing, you know. I was doing some research at Lyle Mech, and me and my colleague, okay, good, you people are better than me. And we were looking at how even a small area of placenta is isolated, and then they are looking at genes in that particular area of the placenta. So the doctor who was showing it, you know, all these placentas, people who try to donate it for research, it was there in the freezer in the research lab. And my colleague was 20 years old than me. She has 3 kids. I have one. And when doctor asked me, have you ever had a look at placenta? And we were like, no. So yeah, we were surprised. So now these scientists, they have separated different portions of placenta, and they have looked at, they have documented the data that there are different species. Okay, there are, you know, the current article, if when I was preparing for this lecture, and if I show you my window, there are about 15 articles open up. And there are about 300 to 500 species of bacteria that we have now identified, that they live on our gut and they are also present on the placenta. So there are only some of them that are written here. Firmicutes and Bacteroidetes. I really don't know how to pronounce them correctly also. Okay, so this is huge. We are kind of sitting on a verge of a, you know, major discoveries into our understanding of who we are. So now let's talk about babies. Okay, so at birth, the GI tract is rapidly colonized with the life events such as illness, antibiotic treatments, and changes in diet, causing chaotic shifts in the microbiota.
You seriously don't know where your tie is, do you?
Sorry,
I said school shorts.
to mute yourself? I'm trying to do it at my end. Let's resume. So the first major exposure of newborns to microbes happened during the birthing process, and it is highly dependent on the mode of delivery. So we now know that if a baby is delivered vaginally, then their skin, gut, nasopharyngeal cavities, they are all enriched with a good symbiotic bacteria known as lactobacillus. And when they compared babies who were born by C-section, those babies' skin, mouth, and gut had majority of microbes known as Staphylococcus, Streptococcus, or Propionibacterium. But does it remain the same? No. So what this figure is trying to tell us is that it can be influenced by various factors. So we talked about birth and then mode of feeding. If the babies, they are dependent on mother's milk, then the colonies in the gut are primarily of bifidobacterium and lactobacillus. However, if there is an early introduction of complementary foods, the colonies change. Okay. And then again, it is dependent on the type of environment the baby thrives in. If there was some infection and they were given any antibiotics, that has an impact on the microbiota. Usually, by the age of 2 to 3 years, every individual have got their own set of colony of bacteria in their gut. But that doesn't mean it remain fixed. It can get influenced by the changes in our lifestyle and in our diet. We all know, the use of postnatal antibiotics can disrupt the delicate ecosystem of this bacteria, these bacteria in the neonatal microbiome. And these antibiotics, they lead to reduction in lactobacillus, and a different set of colonies emerge. And now we know that they can be associated to different disease conditions. So this screenshot is from this research paper by Tamburini et al. And if anybody is interested, I can share it in the chat box. Spend few seconds looking at the summary of research that has been done linking factors that may disrupt microbial homeostasis during early life and development or protection against diseases. So this C-section, this study was done in Danish children, and they looked at the impact of C-section, and what they found was that these babies were susceptible to risk of asthma, IBD, and some immune deficiencies. Then if you look at a study done among Norwegian children, you notice that they were susceptible to certain reactions to foods such as eggs. Then probiotic, okay? So if these babies were given probiotics, then they had a reduction in GI and upper respiratory tract infections. However, if during the ages of 0 to 2 years, if you look at, this was a study done in Finland, in UK, as well as in US, and you can look at the number of participants. There was quite huge numbers. So the data is quite convincing now. The point that I'm trying to make, that if babies have been exposed to antibiotic treatment, then they have the risk of developing asthma, allergy, and even eczema. Okay, so there are various influences across the lifespan, not during the birth time, even after that, that continue to have an impact on the gut microbiota. So let's have some questions. If anybody wants to share anything or want to ask few questions at this stage. So there is one, Alison is asking, is the antibiotic exposure mitigated with use of probiotics? Would anybody like to comment if they have read anything?
Nancy, good morning. It's Kathleen here, Nancy.
Yes, hi, Kathleen.
Hi, Nancy. Nancy, I remember reading articles. I always used to think that probiotics were helpful after antibiotics, but there has been, I think, some studies done saying that they can actually cause the microbiome to take longer to get back to its normal, what it was before the antibiotic was taken, Nancy. But then I was listening to a talk. There's a gentleman, a naturopath in Tasmania, Jason Horolek, the microbiome for years, and he was saying that you've got to take a close look at those studies that were saying that the probiotics weren't helpful. So yeah, so I think, is the jury a bit out on that, Nancy? I'm not sure.
Okay, no, that's a good point, Kathleen, as always. But has anybody else studied anything as well? You want to comment on anything? It's a good question, and Yes, I agree with the presenter that it takes time and that depends upon individual to individual. What is the status of their epithelial layer? And it helps, like in my understanding, give at least 3 months for really bad cases. And probiotics, along with some dietary and lifestyle changes, does help in changing the colony of bacteria. And we'll understand, how that happens when we look at some of the diagrams that are coming up in the slides next. So absolutely, I love these probiotics. So go after your favorite fermented foods because that also help. But as I mentioned, it will depend upon individual to individual. So as usual, let's talk about a story of mine. So I met a person who is now in his teens, but okay, little bit of history. So when he was born, C-section, he had pneumonia, was kept on a lot of antibiotics, low birth weight, continued for about 2.5 years, tried to come back to normal foods, relapse of pneumonia, then switched to some homeopathic medicine, which actually flared the condition. Maybe it was not given in the right dosage or right time. And now the condition is really worse. Eczema, skin changes, terrible, had to be hospitalized. And then somebody told his parents to go after, what are those little ones, the yogurts? The name has completely.
Nancy, is it Yakult? Is that what you?
Yes, thank
Yeah.
you. Thank you, Kathleen. Yes. And somebody told them about Yakult and they started taking it and they could see the changes within few weeks. So when they came to me, he was little better. But I'm like, I don't like Yakult in that sense because it has little bit of sugar. But do you think I should interfere? Should I switch them completely from Yakult to probiotic supplements? What do you say?
Nancy, I think perhaps you could let them know that there perhaps are better sources of probiotics.
Yes, because that baby has gone through so much. I didn't want to interfere and I could see the faith that, this has really helped them. So I said it's okay to continue taking them, but take other things as well. Think about glutamine, think about probiotics, think about selenium, because I am getting a history that his lungs are weak. And remember, I told you in every lecture, I'll talk about glutathione. So that's the endogenous antioxidant to protect our lungs. So I added few things and I did add a probiotic supplement as well, but I didn't say no to Yakult. Okay, so yes, we can reverse the impact of antibiotics. So for this person, it took few weeks, but for some people, it may take up to three months. Okay, so there are some other impacts that you can see here in this diagram. Stress. Stress also plays a large role because, the neurotransmitters, they can influence the gut microbiome by sending stress hormones to the gut via vagus nerve. And then there is another aspect. When we are psychologically stressed or depressed, we may shift, our dietary choices change. Sometimes we go for highly palatable foods, sugars, chocolates, in my case. And the type of food that we are consuming, we are also feeding the bacteria in the gut. So in that sense, we can shift the homeostasis, a balance between good and the bad bacteria. Okay, but our aim is to have the healthy microbiota in more numbers, in more variety. Okay.
Nancy, also with stress, just the way the blood works, isn't it? When we're stressed, blood is diverted away from our gut. So I assume that would impact as well, Nancy.
Yes, Kathleen, that's a good point too. And yeah, that's why why I'm telling you always not to be stressed with the assessments. just try to take everything slowly, because both chronic and acute stressors, they can shift the bacterial colonies. So moving on, now there is, like, remember there was a human genome project which ended up deciphering of every gene that human DNA has. Similarly, we also have a human microbiome project. So in there, we are looking at various metagenomic analysis of genetic material. So just, a couple of sentences of how do we know which colony is there? So they actually take the fecal sample and they are able to isolate the DNA, and from there they isolate the RNA of those bacterial colonies. And then they multiply that in the lab, in the petri dish, and then extrapolate from that data that this person has these many colonies of this particular species of bacteria. So when I was writing this session, I interacted with Microba, which is coming up. It's quite established genetic testing, microbial testing, and that's why I have got a video as your post-lecture activities. So in future, if you want to use this lab testing technique, go on their education portal and you may learn more. I know there are various others as well, but I had the opportunity to learn only through them. Now I love this diagram because I have been making these epithelial cells in isolation in the first 4 lecture, but here you can really see what we are talking about. So if we have an intact epithelial layer, that means each cell is joined to the next one really tightly, and that's what we now know as tight junctions. Then we allow growth of good bacteria. Then above that, some of these bacteria, they secrete or they assist us in making mucus. So that mucus is additional protective layer for our gut cells, so that any pathogens or any toxins can die off there. Okay. Right, so now coming up to gut microbiota's individual functions, I would encourage you all to remember some of them. I have noticed that when it comes in exams, students are talking about the health benefits, whereas the question is there to ask you about how they function, and the result of that function is prevention of those diseases. So let's talk about how these microbiota support us. So one is that they are keeping our gut epithelial cells happy. And how do they do it? Because when they feed on the fiber, fruits and vegetables, the undigested material that reach the colon, they feed on those fibers and then they produce something called as short chain fatty acid. So what's fascinating about that? Now these short chain fatty acid are actually fuel for amazing intestinal cell. So if they are getting it, they are energetic, they grow well, and they are able to have more integrity. Second important point is that they contribute to the maturation of the immune system. So this microbial factory actually supports the largest lymphoid tissue in the human body, which is in the gut, and it's called as GALT, so gut associated lymphoid tissue. So because they both live in the same area of our gut, they support each other. They stimulate local and systemic immune responses, leading to a state of low degree of physiological inflammation. This low degree, remember the word that it's saying that it controlled amount, this inflammation is actually protective against pathogens. So they are fighting with pathogens so that their numbers are low, and the healthy bacteria, they have a good number, good variety of strains. But that all depends on the type of food that we are eating to feed that bacteria. Okay. So what I was saying, Alison, was that these short chain fatty acid, they feed the intestinal epithelial layer, the gut cells, and they keep them happy. Then there are more benefits. And I'm sure some of you would be able to share some of these nutrients that gut microbiome can produce. Any reflections from previous lectures, or you know that bacteria can help producing some nutrients or vitamins in the gut?
Nancy, I know they help with vitamin K. Yes.
Yes, correct, Kathleen. Yes, Tracy, you are right. B12 as well. Some minerals also, you know. Why? Because all these micro minerals, iron, calcium, magnesium, they are all present in the cell wall of fruits and veggies. And sometimes we are not able to digest it. But when that food reaches the colon, bacteria can break that cell wall and release some of those minerals, which may be locally available for absorption. Okay. Right. And then another important point is that there is this competition going on between the healthy microbiome and the pathogens. So if we are feeding the healthy microbiota, it will keep that pathogens away. So some diseases away. Okay, I've just added this screenshot from this article. Again, a very good article about important functions of gut microbiota. So again, I'll remember to share that one when we go on break. I'll share it in the chat box. So most of these articles are full articles and they are freely available. So they are also producing arginine and glutamine. They are helping in deconjugation of the bile acids. They are also inducing regulatory T cells. So again, because they are residing in the same area where our body's lymphoid tissue is, they are able to modulate what form these T cells take. Because, you know, T cells, I might have mentioned this earlier, they are the fighters, but they have to differentiate between the type of fighter they want to become. And microbiome, they support it in that sense. So more functions now. There is recent evidence that suggests that gut microbiota affects various metabolic pathways also. So the understanding is that they may be regulating weight somehow. And a different colony of bacteria may be responsible for development of obesity. And there was a very small study which was done by a group of scientists known as Jumperds et al. So they did an energy balance study in 12 lean subjects and 9 obese individuals. So they gave them, you know, calorically distinct diets for very short duration, about 3 weeks. And then they looked at the gut microbiota by doing all those metagenomic studies. And what they found was that there was a rapid difference in the gut microbiota of the people who consumed high calorie versus low calorie. So the people actually had more of firmicutes, okay, rather than bacteroidetes. So I don't want you to remember all these names, but the point is that there is a strong link between gut microbiome composition, nutrient absorption in humans, and its metabolic impact. Then we also know that some of these bacteria help in metabolizing histamine. So, you know, some people have histamine allergies. So now they have found that it needs a particular enzyme, histamine decarboxylase. So some people may not have that enzyme. That is the reason they may be having allergy. I'm simplifying it. It is a little more complicated with that. So now they have found out that some of these bacteria, they can produce histamine decarboxylase enzymes, and that help in metabolizing histamine so that it does not interfere with somebody's metabolic function. Then another one is GABA. You know, they are saying that there is a crosstalk between our brain or our neurotransmitter and these bacteria. Because when they are doing their own thing, they are feeding on fiber and they are producing these short chain fatty acids, and we are coming to their names in the next slide. They also produce GABA. So what are these short chain fatty acids? These are produced by bacterial fermentation within the large intestine from undigested carbohydrates. And the names are acetate, propionate, and butyrate. And they individually, they have got various benefits. One is if we have more of SCFAs, that changes the acidic milieu of the lumen that we saw in that previous diagram. And I mentioned that they are key energy source for colon cells. They also activate mucin release so that the cell layer remain protected. And they may also increase mucosal blood supply. Okay, questions and comments. So Juliana, you are right. You had that answer ready even before we reached this slide, that SCFAs are butyrate, propionate, and acetate. So Alison is asking which bacteria help with this. So you're meaning generation of SCFAs or the other benefits that we talked about in the previous slide?
So that question was with regard to the histamine allergy. You talked about certain bacteria producing components or helping produce the histamine decarboxylase.
Yeah, very good question, Alison. So one constituent that I can recall is Lactobacillus reuteri. I'll write it down because kind of I might not be pronouncing it correctly.
Oh, thank you. Nancy, can I also ask, are you aware of a gut microbiome that might affect motion sickness?
Oh, no, not really, Alison. But,
Okay.
but yeah, sorry. But we can look at it. I'll have a look. Okay. I'm just writing it down so that I don't forget. Any other comments or questions? Okay, Tracy is asking if histamine can be regulated with gut microbiome. Would someone who is prone to rhinitis benefit from probiotics? What do you say, guys? Tracy's question. Yes, Courtney, definitely. Yes, it would. Okay. So yes, that's what we are reaching, that yes, it can. Okay, but we are not talking about the individual clinical therapeutics today. That you will learn when you study NMDC 221, your clinical subject. So today we'll continue with what they really do, and we'll talk about only a couple of them that have sufficient evidence to prevent diseases. But it's interesting, isn't it? Whenever I'm reading about it, I sometimes think like, are we really alien? Because now we know it's not only bacteria, we have some good fungi that are fungi that are living in the gut, and they are really supportive. So again, one of the stories that when we were growing up, and I'm talking about India, and we had any gut issues, and if the GP was giving me an antibiotic, they would also ask me to take probiotics. Even today, and one of the components of those probiotics, one would be lactobacillus, and then we make our own yogurt at home. So that one has a lot of lactobacillus sporogenes. So that's a different colony altogether. And when I go and look at the yogurts that are sold in the supermarkets here, they have mainly lactobacillus rhamnosus. So that's the main one. And it is a really good bacteria to have in the gut and in the food. So another component of those probiotic would be Saccharomyces or Saccharomyces again, my pronunciation, boulevardi. So that's the one. Look out for that in the supplement. Combination of Lactobacillus rhamnosus, Bifidobacteria lactis, and it will come up later in the slide. So don't bother about the spelling right now. And Saccharomyces boulevardi. Okay, so now we have found out that there is a crosstalk happening between our organ systems. And what we know now is, first one is gut-liver axis. So when we say gut-liver, that means gut has microbiome and they can interact with the liver. They can interact with the metabolites that are coming from liver and in turn get impacted by the liver functioning. Okay, so this is a bi-directional communication which is happening between our gut and liver. I should also point out here that please download this article by Tripathi et al. It's given in the reference list. This one will help you in your assessment too. So all the information regarding your second assessment is in this article. Now, how is this communication happening? It's happening through biliary tract, portal vein, and systemic circulation. So, you know, sometime if I'm consuming a lot of red meats, okay, so it comes for digestion, and gut bacteria are able to convert it into TMA. And I have the full form written somewhere, I'll tell you that. So it's mainly the choline in those red meats that is converted to TMA by the gut bacteria. Then that TMA reaches the liver, and liver has to excrete it, and liver converts it into TMAO. If there is an issue with liver, somebody has fatty liver, then there is excess of TMAO circulating in the blood because it cannot be excreted because liver is sick. So we need those healthy bacteria at the first stage to convert it into TMAO. So that's one aspect. And again, we are changing the type of bacteria in the gut by the type of proteins that we are eating. Now, if there are issues with the gut, because they both are connected, and let's say that there is dysbiosis happening or intestinal layer is disintegrated, then these bacterial metabolites, MAPs, again, we'll come to the full form in the next slide, they travel to the liver. Now, these are foreign bodies for the liver. Liver becomes inflammatory. It tries to fight off these PAMs. And in doing so, it increases inflammation. So there are cytokines moving around, mainly tumor necrosis factor alpha, TNF. And it continues. Then those inflammatory molecules travel to gut and further harm the bacteria, and so on and so on. Now, what's written here is acetaldehyde, butyrate, and ethanol. So again, it's an indication of what we are eating and what we are producing in the gut. If I'm going after alcohol every day, then there might be excess of acetaldehyde, which may be harmful to the gut bacteria, which may also put burden on the liver. However, if I'm consuming a lot of resistant starches, then my gut bacteria would be producing more of butyrate, which actually is protective against inflammation and all other degenerative changes that our body may be happening. Okay, so it's kind of the same thing that's now written in the slides. The explanation about that figure about liver and gut axis. So just reading the first sentence here from the slide, that intestinal factors regulate bile acid synthesis, glucose and lipid metabolism in the liver, as the intestinal microbiome generated products, which are microbe-associated molecular patterns, may travel via portal vein and influence liver function. And you know, there are other axes. Now we know that there is a gut-heart axis, gut-kidney axis, and gut-brain axis. So remember, elders in our family always used to say, I have a gut feeling, I wonder uh, they were referring to that gut-brain axis or what? Again, another funny story. So whenever I had to go for an exam or an interview, when I was younger, my mom would come to me with just 1 teaspoon of yogurt, homemade yogurt. And she'll say, have it before you go for exam or anything. And it still happens there. And I am fighting with her. Like, what does it has to do? I want to give exam empty stomach. So I don't even want to have that 1 teaspoon of yogurt. And she said, no, just take it. And whenever I'm interacting with you all in this session on microbiome, I always wonder, she never knew that that's going to feed the healthy microbiota, or it's going to have that probiotic effect, and that there would be a gut brain communication happening so that I remain calm during my exam. Okay.
Nancy,
Yeah.
can I just tell you too, when you chat about the bile acid synthesis, I was listening recently to this gentleman. He's very good, Jason Horolik. He studied the microbiome for years. He was just chatting how there are, like if we eat, I guess, high fat diets, there's bile acid loving species within the gut. And I think it's hydrogen sulfide producing species.
Yes.
They love bile, so then they build up and that creates problems, Nancy. So it's just sometimes can be a vicious circle.
Yes, agreed, definitely. And thanks for sharing that, Kathleen. Would anybody else like to comment or share anything? Any question regarding gut-liver axis? So the point is that whatever we are eating, it is feeding that microbiome, and it may influence their colonies and species. So what happens if there is dysbiosis? So dysbiosis is a state where there is a change in that commensal to pathogen numbers in the gut. So remember that definition also, that there may be more of pathogens and less of those normal symbiotic bacteria living in the gut. So that leads to gut dysbiosis. Then you might have also heard about small intestinal bacterial overgrowth. Now, this is the presence of excessive bacteria in the small intestine, and it is implicated in the cause of chronic diarrhea and malabsorption. So the point is they have to stay in the colon. And if for some reason there are more of pathogens, there are more numbers, and they are interfering with the normal mechanism of the body in the small intestine, that can be an issue. So I'm sure most of you would be aware that gut dysbiosis and even SIBO can have some symptoms such as bloating, abdominal distension, diarrhea, fatigue, weakness. And we are talking about, superlative degree of fatigue and abdominal pain. Now, clinical manifestation, if we want to confirm whether this is dysbiosis, then we also test for fatty liver, weight loss, some mineral deficiencies, such as iron, vitamin, and B12, before doing specific SIBO test. So in that, has anybody heard about those testing? How to test?
Nancy, it's a breath test, isn't it, with the glucose, lactulose, and sometimes the fructose?
Yes, very good, Kathleen. Yes, that's correct. So we have those tests in place. Thanks, Michelle. Yes, you're right, a breath test. So we give a particular nutrient, as Kathleen is mentioning about glucose, and then we measure the metabolites in the breath. Through that we can measure that whether they have a particular malabsorption, which may be now related to particular colonies. Okay, so now we know how important microbiome are. We are aware about some of the names of good bacteria. So if there is dysbiosis, it is now correlated with changes in the gut microbiome that is linked to ulcerative colitis, Crohn's disease, irritable bowel syndrome, and even colon cancer. So let's have a look how it is happening. So on your left side is this diagram showing about when there are more of pathogenic bacteria. So can you see that gut layer, those gap junctions, or I talk about those tight junctions, there is a gap between them. And remember what's on the other side next to the basolateral layer are the blood vessels. And there is the lymph. So it would have all your immune cells. So whenever there is this gap in the tight junctions, our immune system gets threatened and they start causing inflammation. So their intentions are good. They want to fight away any pathogens that may be coming up. But in doing so, they even start harming the epithelial cell. Okay. So that's the reason now we are associating dysbiosis with inflammatory condition. I have been asked whenever I discuss this slide, does gluten has a role there? Is that the reason? Or is it not gluten? It is the way we are farming wheat that may be a issue here. Would anybody like to comment? Has anybody read anything or faced anything like that? Do you think that could be an issue?
Nancy, sorry, could you just say that again? I'm so sorry, Nancy. What were you asking?
Yeah. What I was saying was that whenever I discuss this slide, I have been always asked whether gluten Okay.
There, Nancy, you're back.
Can you hear me now?
Yes, Nancy.
Oh, sorry about that. Sorry. I don't know why that happened. Sometimes I get a message, your internet connection is unstable. Okay. So we are back. That's good. So Michelle is saying, yes, gluten has been shown to loosen the tight junctions. Yes, absolutely correct, Michelle. Yes, so we have that evidence now. But again, the other aspect that I'm asking is that is it gluten or is it the way we are farming wheat that is adding to the issue, or maybe that is causing the issue? Yes, Allison, might be glycophosphate. So that's my understanding. And again, it's all connected, isn't it? Maybe somebody's gut is weak because they have been exposed to a lot of antibiotics. And that causes them to have less tight junction, so their epithelial layer is susceptible to even minute exposure to such environmental toxins.
Nancy, so can I just ask, so with the gluten, you're actually saying it's not the gluten, it's the sprays or the way we go about farming? Is that what you're
That's
saying?
one understanding. That's one understanding. And second, it may be that those people may have a leaky gut. You know, we call this biases sometimes as leaky gut because the way they have been fed in their early years, that has led to lack of these tight junction. So any even small toxin coming in the gut is leading to that inflammatory cascade. So it can be gluten intolerance. They may have an issue, but majority of the cases, it might be just because of the way. Yeah, it can be both also.
And Nancy, I know it's the butyrate, isn't it, that feeds the enterocyte. So like you said, maybe it's more in if you're lacking those species that produce the butyrate.
Correct. That
Yeah.
could be a reason as well. Yes.
That could
Can I just ask, if it's gluten, if it's not the gluten, if it's the farming process, then you would actually think that if you avoided gluten but had everything else, you'd still have the same issue because the farming processes are still using similar chemicals. Like, I avoid gluten completely, but it's the only thing I avoid.
Okay.
So I'm just sort of, I don't understand if it's about the farming process and it's about some chemical as opposed to gluten, then I would expect that if I have other farmed produce, then I'm still going to be affected with the same issue if it's a chemical thing as opposed to a gluten thing.
Yeah, no, I completely agree. Now, there are 2 points happening. That in the first case, some people may genuinely have gluten intolerance because, again, because we have changed the grain so much, we have changed the chemical structure of things because we have been doing for so long. So we are trying to say something, and one of the reasons they may have gluten tolerance is because those components directly attack the tight junctions. So if they may have leaky gut, that it's kind of a what came first, chicken or the egg? You know, it's kind of that issue. They may have gluten intolerance because they have loose intestinal cells, and that exacerbates their gluten intolerance response. However, if we can target the gut, we can give them some glutamine, we give them some probiotic and restore that gut epithelium, provide them an integral layer. In the long term, I'm saying again, in the long term, it's a long work to do with people. And again, the point that Catherine was saying, we change the microbial colony that is able to support in metabolizing gluten, it can be resolved. Okay. Did I answer you, or am I still confusing you?
You're not confusing me, Nancy. You're not doing that at all.
Yeah, but I
no, it's a
completely
fine
understand.
explanation though, yeah.
We are trying to all find answers. We are trying to understand. So yes, we can address that, but it takes a long time to repair that damage. Interesting, isn't it? Okay. Couple of slides about impact of dysbiosis on other disease condition, and then we'll take a 10 minutes break. So now gut dysbiosis may induce a low-grade inflammatory activation of macrophages, and it may lead to even diabetes, obesity, metabolic syndrome. And one of the mechanisms that we have learned is that in dysbiosis, when we have more of pathogens, they secrete this inflammatory molecule called as lipopolysaccharide. So when we look at the fecal sample of people who have type 2 diabetes or obese, this has been confirmed in human study as well. I have taken this from a mice study, but it has been confirmed in human study that those fecal sample would have more of lipopolysaccharide, telling us that they have some pathogens in their guts. So that's just one aspect. There are other aspects that if we are feeding more of bad fats, or we don't have a balance of good and the other saturated fats, then also we change the microbial colony, and by their metabolites they can influence a person's hunger, satiety levels. They can induce insulin resistance. So that's how close the interaction is between the microbiome and metabolic conditions.
Nancy, can I just ask with regards to the insulin resistance in that particular study, the study written down there, is that the mice study or is that the human study? Or there's actually two written there, sorry.
Yes, there is a two. I think Fabiano is the review article.
Right.
This information is coming from the mice study. Yes.
Okay. Thank you.
Yes. Okay, so then coming up to dysbiosis and mental health. So when I was saying that gut talks to the brain. So for a long time, researchers had tried to understand how that may be happening. And now we know that it may be via the metabolites that these bacteria are secreting or producing. So under conditions of stress, there is HPA axis that modulate cortisols, and then cortisol can impact immune cells. It can also alter the gut permeability and the barrier function. This all kind of lead to change in the gut microbiota composition. So now we have a different microbiota. They are not producing GABA. They are producing some other toxins that are interfering with the connection. Okay, and it's again, whenever I'm sure you might have heard sometime when we are stressed, we may have loose stools. And some people, you know, when they are traveling, again, they are anxious, they say that we are constipated. So in a way, there is two-way communication happening. Okay. So the latest research that is coming up, it is saying that gut microbiome imbalance has been associated even with depression, epilepsy, and autism as well. So this is quite recent, but it's a whole new world. Okay. So it is again an explanation about that liver-gut axis, that when there is a dysbiosis, then even that gut-liver axis is deranged. Okay, let's take a 10 minutes breaks. Okay. And yeah, we had all the good discussion even during the break time.
And Nancy, will you post those articles too later when you have time?
Yeah. I have a whole list of them open.
Thank you.
And I'll definitely post them. Oh, I was supposed to do that during break time.
And Nancy, can I just say one other thing too? You know, when you chat about pathogenic bacteria,
yeah.
understanding is within the gut, Nancy, that any bacteria can become pathogenic if it becomes if it doesn't stay in symbiosis. So if conditions become conducive, that bacteria can multiply and become dominant, I guess,
Yeah.
that can become a pathogenic bacteria. But yet when it's in its normal levels, it's a perfectly fine bacteria.
Yes.
So, what I've learned, sorry, Nancy, I'll let you go with that.
No, that's fine. You are correct here again. That's why we keep saying balance. Our body is all about maintaining a balance. That's why we are talking about so many colonies, because they keep each other in check. So even the good one and even the bad one, they are themselves kind of a complete, I would say, a planet. So yes, they keep themselves check. Okay, I'm thinking. Remind me again to post those articles, Kathleen, before.
Yeah, I
end
will, Nancy.
today.
Thank you.
And I'll post it on the chat box. Okay, let's move on to probiotics. Sorry, I don't think we had any break, but we were having such interesting discussion. So now, how do we correct dysbiosis? We learn about some probiotics and prebiotics. So literally the word probiotic means pro-life, and we define them as live microorganisms which, when administered in adequate amounts, confer a health benefit on the host. And I was talking about various species. Here is the list of some of the popular one. So if you have a client with eczema, trying to think about Saccharomyces boulardii and acidophilus. Then bifidobacteria is also very good if somebody has any allergic response. Reuteri is the one which I was talking earlier, the histamine one, okay? And then Rhamnosus GG. So Rhamnosus is the species, and GG means it has the same gene. So that one is really beneficial for eczema.
Nancy, what do you mean by Rhamnosus GG has the same gene? What do you mean by that? Sorry.
So we have two,
Rhamnosus. Yes.
Yes, Rhamnosus GG is the name of the strain that it has, on the X chromosome, like we have got two, again, the word is not coming to me now. We can have two translation, it could be different strain. So when we say GG, we have a, that word is not coming to me, Kathleen, sorry.
That's all right, Nancy. You're okay. You're okay.
I'll come back to it. Okay.
All right. Thank you,
So
Nancy.
many technical terms and
Thank you. It's early. It's all right. Thank you, Nancy.
You are too kind, Kathleen. It will come to me and I'll tell you. Okay. So now we have bifidobacteria, lactobacilli, and thermophilus. They have been most recognized probiotics because they can survive the upper GI tract. They can proliferate. They can colonize themselves. And we now know that they can address clients who have diarrhea, people with lactose intolerance. They can strengthen the gut impermeability. They can even address dermatitis. Okay. So, important point to remember from exam perspective, which I always like to share, is mechanisms of action of probiotics. So number one, They are able to support biosynthesis of vitamins and even some amino acids. They produce SCFA, butyrate propionate acetate, so that they are fueling our intestinal cell. Again, I like this diagram because it is showing how we have a thicker mucus if we have probiotics. The epithelial layer is strengthened. These probiotics, they inhibit pathogen adhesion. That means they don't allow them to get attached anywhere with the help of maybe the metabolites that they may be producing, or simply because they are too much in numbers that they dislodge the pathogenic bacteria. And then they are able to talk to the immune cells as well and have a balance of Th1 and Th2, which is the T helper cell 1 and 2. Again, something extra, that there is now a lot of data that the allergy or autoimmune responses because of imbalance between Th1 and Th2. So again, something you will explore in your further subjects, clinical subjects. So in this slide, apologies, I have made everything red because these are the points that I would like you to remember from the exam perspective. So I know you all understand how these probiotics are assisting the gut, what are their functions, but sometime in the exam scenario when we are stressed, some of these words, they leave us. The way some words failed me today while I wanted to answer Kathleen. So remember some of these steps. Some individual benefits. Okay, tell me at this stage some of your favorite fermented foods.
Well, Nancy, I'm very excited. Yesterday I got some kefir grains, some milk kefir grains. So I'm sitting on my bench at the moment. They have to, sit for 24 hours, but I'm excited to try them, Nancy.
Yeah, that's awesome. Yeah, good. Sauerkraut. Yes, Mary, I love that too. Again, it was, I was introduced to it from one of my students in one of the sessions. We all cooked something and we shared it, and she made a sauerkraut for me. So I still remember. Yes, Michelle, kimchi is another one.
Nancy, I've tried beet kvass as well. We learnt to make that as well yesterday, and that was lovely. That's very easy to make, but it was. I've had it before, and it's lovely.
Good. Send me some of your recipes, okay?
Okay, Nancy. Nancy, I'm happy. I know I asked you about kefir grains, but when they grow a bit, if you want some, I'm happy. I'll bring some into you.
Yes, definitely. We'll share something. We have to be, doing that.
Yes, and if anyone else, I don't know if anyone else lives in Adelaide. Yeah.
Everyone, catch hold of Kathleen. Good. Okay, kombucha is another one, isn't it? One of my students gifted me that Scooby. Yes, and she gave me a few recipes to try it with some strawberries, that was a good experience as well for me. Okay, so now apart from probiotics, we also have prebiotics. So we may give somebody probiotics, supplements, 3 months, 4 months, but if we are not supporting them with the food that these bacteria love, it is all going to be useless. So there are a few definitions for you of prebiotics. Then different kind of, I'll come to bifidobacteria in a while. So there is a criteria that is being used, and it is there is a consensus that they have to be resistant to acidic pH. They have to be fermentable. So microbiota should feed on it, otherwise they're useless. Okay. And then now we have some of the examples. Mainly we talk about fiber, but all prebiotics are fiber. However, not all fiber are prebiotics. That means not all the roughage that we are eating is supporting the bacteria. Good bacteria, I should mention. And again, that's based on our understanding so far. So when we say, what we now know is that butyrate, as Kathleen was mentioning earlier, is the most known to most researched short chain fatty acid. And why? Because butyrate is produced by bifidobacteria. That's our understanding that this is the one. Okay. So they have a preference actually for oligosaccharides, and they produce a lot of butyrate. Okay. And that too, more and more research is coming that it's the resistant starches that are so important. So fructooligosaccharides, they are bifidogenic. So when I say bifidogenic means that they are feeding bifidobacteria. And then we have some of the examples of these resistant starches. So one of my favorite is plantain or the green banana. I have a recipe from my mom and I'm the only one who love it in my family. So yeah, I make it occasionally, and I even make a vegetable with banana peel. So when they talk about sustainability and everything, so that banana peel, I make it with the peel of ripe banana. It's an awesome recipe.
Nancy, what do you do with the green banana?
Yeah, I make a veggie out of it. So I peel it and I just saute it in some of these spices.
Wow.
It's yum.
Yeah, do you think you might share that recipe at some point?
Yeah, definitely. We have a lot of things to share this.
Yeah, we do.
Yeah. So anybody who likes resistant starches or has any recipes, feel free to share with all of us. Okay, good. I think there's a lot of discussion going around those probiotic and prebiotics. Yes, Tracy, I use normal unripe bananas. Yeah. I've retained this slide just to see which fibers are acted upon by the gut bacteria. So you can see resistant starches have 100% fermentability. Wheat bran is good, and guar gum. Okay, what do we know about guar gum?
Nancy, I take a prebiotic, just a bit of powder, the partially hydrogenized guar gum. I've only just started it recently. It's safe if you've got like a small intestinal bowel overgrowth, apparently.
Yeah.
Builds bifidobacterium, and I think I think it could I think it might be lactobacillus as well. But yeah, it's anyway, Nancy.
That's fine. Does anybody else has to share? What are they taking?
Yeah, I also take the same as you, Kathleen, I think, the powdered dry form.
Yes.
Yeah. See, again, it depends upon how our body tolerates it. What is the RDI for fiber, guys?
Nancy, it's between 20 and 30 grams?
Yeah.
That's what they say the RDI is, but they say it's like our ancestors used to eat, I think, about 100, didn't they? They said 100 grams or something of fiber a day. So what we take is very small, the RDI.
Yes.
Yeah.
But again, it will vary, isn't it? Because some people who is coming to me with a lot of gut permeability issue, and I suspect that the colon cells are not happy. So after correcting that phase, I may up their intake of fiber a lot more than just simply 20-25 grams. But I have to wait till they have a strong gut. Thanks, Michelle. Yes, it's 25 gram for women and 38 gram for men. Okay, I'm just going to go into the fridge and I'm going to show you guar gum. Okay, just bear with me. You know I do all this stuff. Just okay, guys. So I know it might not be a good view, but we actually have this vegetable known as guar gum. Okay, so I buy it from an Indian supermarket and it's frozen because it's not grown here. So these are just like beans, you know. And so yeah, I love this veg. So that's my source of guar gum.
So Nancy, does that naturally contain guar gum, these
Yes.
beans?
Yes.
And can you grow them here in Australia, Nancy? Do you know?
Yeah, I have shared you that I have a long list for my husband to try.
Yes.
All so definitely we try. Haven't tried yet.
Tried it. Well, let us know if he's successful, Nancy.
Definitely. Okay. Okay. There was a question in the chat box regarding what is fagula. Not sure. I should have a look. I know something else called as isabgol. I think it is that only. It's a kind of a fiber. Yeah. Have to find that picture as well for you all. It's a white, thick grain. Okay. So, yes, Mary, I cook guar simply with those spices and little bit of mustard oil. It takes a longer to cook, but don't overcook because we want that fiber not to get hydrolyzed. So simple sauteing would do. And I have to find you what isabgol is. Okay. So one more thing. So these are some of the benefits of fermentable fiber. Again, try to remember some of them, that they act as prebiotics. So they assist in generation of short chain fatty acid because they are feeding the gut bacteria. They help in mucosal cell differentiation and proliferation. And they also help in having a good acid environment in the gut. Okay, so just again, another beautiful picture showing what are the health benefits of fibers, and in a way also helping us understand how prebiotic and probiotics, they work together. Okay, so when we are having a fiber-rich diet, you can see the red ones are the mucus degrading microbiota. So their number is controlled, and we have more of fiber degrading microbiota, which is also ensuring that there is more mucin that protects our gut layer. And it is the reverse. If we are not on a lot of fiber and we are going crazy after processed meats and other processed food.
Nancy, can I just say too, I don't know if you have read about this, but they also chat about how fasting is beneficial for the gut bacteria. You know, I think 12 to 15 hours, because some bacteria, they actually feed on that mucin layer. So if we do a bit of fasting, we build up those bacteria as well as the ones that feed on the fiber. But I don't know if you've heard about that, but that must be very beneficial for gut health as well.
Yes, Kathleen, you may be right. I have not read about it.
Mm.
There's
Just
so much to
increases
read about,
diversity. Just
but it
increases
does
diversity.
make sense, isn't it?
Yeah. Yes, it
It
does.
makes
It does. Yeah.
Now, last couple of slides, we are talking about how we need to have a balance of both because there is a camaraderie going on between prebiotics and probiotics. Okay, so we can use probiotic in different disease conditions. And when there is fermentation of polyphenols happening, we are stimulating production of bifidobacteria, and there is a change in the ratio of firmicutes and bacteroidetes. So some repetitions here as well in the slide, but the aim is to understand that they both help each other, and the end result may be that they protect us from certain disease conditions. Okay, so again, the summary of how they bring about probiotic impact. In fact, people who may have excess of Helicobacter pylori, so now they are saying that it has been there for ages with us through the entire evolutionary process, but it has been kept check with other bacteria. So people who may have acid reflux or tendency to have ulcers or issue with H. pylori, even that can be addressed by having a balance of other probiotic species of bacteria. So one last thing from the exam perspective, that we now have human studies that have shown if the mothers are given Lactobacillus rhamnosus strain GG prenatally to the mothers, and also during the first few months when the baby is born, and we know they have a high risk of atopy, then prevalence of atopy and even eczema can be reduced. The second evidence that we have is regarding Bifidobacterium lactis, so BB12. That one also supports reduction of eczema in children. Okay, that's all for today. So the plan is that I kind of divide you in different groups so you can talk about the articles that I'm sharing with you, have some interaction there, and then we'll come back in the last 5 minutes and answer if you have any questions or summary of the discussion that you have while discussing that. So at this stage, we are discussing this pre-lecture reading in groups for the people who are listening to the recording. So this paper can be assessed from your LMS. It's one of the pre-lecture reading on influence of diet on gut microbiome and implications for human health. It will be a good idea to at least go through one of the discussion points. So it talks about how consuming different kinds of fats changes the gut microbiome. Or it also talks about how different carbohydrates modify the gut microbiome. So, and then fiber also, and what are the mechanisms and which are the species of bacteria that are enabling all those benefits. So take some time to read through and make your own summary notes. Hi everyone, welcome back.
Hello again.
How was it? What did you discuss?
So we were really interested in the in that paragraph where it spoke about the benefits of lactose.
Yeah.
In this page, yeah, just there at the bottom, lactose is commonly thought of as a potential gastrointestinal irritant, but it said just above that it has been observed to increase the concentration of short-chain fatty acids.
Yeah. So can we apply it for some of our clients, this information? Do you think this is sufficient information?
No, because we don't know if they, it depends if the client has a lactose intolerance or not.
Yeah, it is based on, I think, only one study, isn't it? If you go back and look at this one and this one. Yeah.
Okay.
Yeah.
Yeah.
So that's always a good idea to go back and look what kind of research they are quoting. So, any of the sugars had any beneficial effects with respect to microbiome? Again, that was my study. So I would like to learn more about this one. Any other new information or any other observation that you had? And kind of did it reinforce what we were studying in the session today?
Yes, Nancy, I felt it did.
Okay, so okay. Thanks, Michelle and group. And would anybody else like to comment? The other group? What about the fiber? What did you find? Any new information?
Nancy, I don't know if Jane or Charlie want to say. We had Julian as well, but Julian didn't chat at all. So I don't know, Nancy, if Julian was there, but if Julian wants to say something too. But yeah, we just chatted it reinforced, Nancy, what we studied in the lecture. That, the studies has found that, high intake of prebiotics, they mentioned what they are up the top there. It increased the gene richness, the bifidobacteria and the lactobacillic acid. And we just chatted about how, Nancy, if we take a variety of things, I mean, they say you should eat about 40 different vegetables a week. You're going to feed different species, increase your diversity. And they say that the more diverse your microbiome, the better your health. So we felt that article was very much it just reinforced what we'd studied today, Nancy, and what we've heard. But I don't know if Jane and Charlie want to add something to that.
Yeah, feel free to add or discuss anything or even questions at this stage. Thanks, Kathleen.
I'd agree with Kathleen. We sort of just talked about, it mentions how quickly gut bacteria can change. We sort of talked about the consistency of eating fiber. So if you don't eat it for a few days, how impactful that may be on your gut bacteria and how quickly it can alter.
Yes. Yeah. Okay, then, based on all this, tell me if you have a client in future and you notice that they have gut permeability, leaky gut. Okay, so yes, you want to give them probiotic. As we were discussing earlier also, and as Jane and Kathleen, you're also saying that the shift can happen quite quickly. So would you like to recommend this client with leaky gut prebiotics in the form of fresh salads along with the probiotics in the very first consult?
Nancy, I would have to say that experience, Nancy, that sometimes your gut is very inflamed and damaged. And that sometimes you cannot, those foods are beautiful, but just sometimes until you heal it, you cannot tolerate them, Nancy. So I think you have to tread with caution with just slow changes, Nancy, is probably what I would say.
Yes, I agree with you, Kathleen, that most of the time when people are coming to us, they have a very fragile gut and it's a long process. So, but we want to give them some support in the very first consult, so their probiotics help. However, you know, we can't change the dietary style in just 1 week. It takes time. So maybe trying to give some zinc and glutamine to support the gut healing for about a month, then intervene with some prebiotics. So because actually it can give them more bloating, it can increase their pain if we kind of prescribe them a high fiber diet in the beginning. So that's my understanding as well. And somebody was asking what is Isabgol. So I found out that psyllium. That's the brand name for psyllium. Okay. And regarding that answer about what is GG, I thought that it has something to do with the gene expression, but it's not. And I will post all this information on Loop because some of you have left the discussion. It's actually the initials for the first person through which this gene was isolated. So thanks for asking your question. I learned something new today. So that's all for today. And I didn't find anything on motion sickness specifically, though we can make speculations based on our understanding and experiments, but nothing in research yet. But I'll keep looking till I do that post.
Nancy, and I'm so sorry, I can't see those articles in the chat box anymore. So I don't know, would you be able to just post them when you send us your bits of it, or can we get them from the reference list, Nancy?
You know, what I found was one of that article is no longer freely available.
Okay.
That's why I made it a point that I should post it to you. So let me try it again. And you know, when I try to post multiple things on Loop, it doesn't allow me.
Okay. They were there, they seem to be sitting there as a PDF, but when we went off into groups, Nancy, they've gone.
Okay, let's see now whether you can download it. Otherwise, I'll have to.
Oh, I'll try, Nancy. I've just brought one up now. I'll let you know, Nancy. It's just downloading it as we speak.
Yeah.
And I'll just go and see if I can.
Mine's done the first one.
Okay, thanks.
And also, before you go, last week you mentioned a webinar tomorrow regarding the assessment. Are you still planning on doing that?
Yes, so I posted something on the subject notice board. I have posted the link. We are doing it on Wednesday, 31st March. Okay, so did you see that subject notice?
I actually haven't checked, so I'll go in and have a look.
Yes. Has anybody else seen it? I'm also looking it again that the
Nancy, I saw it. It came through to me. Yes.
Okay, yes. Because sometimes we have to select all the participants, and the first time I forgot it. So I reposted it so that everybody gets it. And I'll be recording that webinar. It would be available from the very next day in case you can't attend it.
And the next Monday, on Easter Monday, we're just to watch, attend another lecture or watch recording?
Regarding the assessment?
No, for our lecture on Easter Monday.
Yes, so the thing is that for Easter Monday, no Monday classes, but you are free to attend Tuesday or Thursday morning or evening lectures, and I'll be recording the Thursday lecture.
Okay.
So no Monday.
And Mansi, do we have a bit of a time change after Easter, don't we?
Yes, there would be for some of us, and EdTech should be posting new links. In fact, that has been playing on my mind since last week, and I'm waiting. They should do it by this week. Otherwise, I'll remind them, because at my end, the links are changing. So you should also be informed about the new links and new timings. So definitely look out for my post on Thursday.
Is that the time change? Is that like relevant for people in New South Wales and Victoria, or is that more like who does that affect? Do you know?
You know how bad I am with daylight saving.
I'm just saying.
It definitely affects Adelaide. I'm not sure about the others.
Okay.
So we will have some information by Thursday.
Perfect. Thank you.
Great. Now have you got the links to articles?
Nancy, yeah, I was able to download them. Thank you very much, Nancy.
Yeah. Thanks, everyone.
Thank you.
Have a good
Thanks
week.
for your time. Have a good week,
Thank
Nancy. See you,
you.
everyone. Thanks, guys. Bye-bye.